Jai Gurudev students. Our next part is ribosome. So regarding ribosome, and this ribosome was first isolated from the cell cytoplasm by the QD in 1943 AD, and these were called ribosomes by G. Palade in 1955 AD. That is the history. And then definition: the ribosome are the smallest known electron microscopic, submicroscopic ribonucleoprotein particles attached either with the rough endoplasmic reticulum or flattening freely in the, it is floating freely within the cytoplasm and at the site of protein synthesis. So definition: an occurrence, it is present both in the prokaryotes and eukaryotes. Now the type, that's 70S ribosome and 80S ribosome. Now what is this 70S? 70S ribosome, that is a sedimentation coefficient. Sedimentation coefficient is a unit. Sedimentation definition, you know about that. Due to centrifugation, the sedimentation takes place, and that coefficient, and 70S one and another one 80S ribosome. These 70S ribosomes are found in the prokaryotes and mitochondria and plastids of eukaryotes. Okay. And in case of the 80S ribosomes, these are found in the cytoplasm of eukaryotes mainly, and this sedimentation coefficient is 80S. This one is that is sedimentation coefficient is 70S, and this one is 80S. Now chemical composition. What is the chemical composition? So ribosome is chemically called ribonucleoprotein RNP particles. And what is polyribosomes or polysomes? When many ribosomes are attached on same messenger RNA strand, it is known as polysomes or polyribosomes. Now see the structure. 70S, this one is 70S, this one is 80S. It doesn't mean that 50 plus 30. Okay. So it is actually, its sedimentation coefficient is 70S. At its sedimentation coefficient together, it is 2 subunit is there, 2 subunit, here also 2 subunits. So coefficient is 80S, and here the coefficient is, sedimentation coefficient is 70S. Okay. And length and breadth is given. So ribosome, this one is present in prokaryotes and this one is mainly present in eukaryotes. Now what is the structure? Smaller unit, that is upper one smaller unit, and another one is larger unit. In both the cases, smaller unit, and separately I have drawn the diagram, base part, head part and clip part. This one is attachment part. And in case of the large subunit, this one is base part, protuberance part, ridge part means attachment part, and stalk part. When these two are united, it looks like this one, smaller unit, and this one is larger unit. So larger unit with the help, that is larger unit with the help of this ridge and protuberance, it is get connected with the smaller one with a clip. So this one is structure of the ribosome. Now what is the function of the ribosomes? Ribosomes are called the protein factory because it can synthesis protein, and protein synthesis takes place within the ribosome. So this is the structure, is a very, very important one. Our next cytoplasmic that is structure is mitochondria. The term mitochondria was given by the C. Benda in 1897, and where it is present and absent, these are absent in prokaryotes, but these are present in eukaryotes. Shape may be oval, discoid, granular, or fibrillar, or spherical. Now number and position. Maximum number of mitochondria is found in, that is flight muscles of the insects. These are more in growing and dividing and metabolically active cells, mitochondria, because all of you know from our previous study that it generates energy. So it is present in the flight muscle more in number. Now what is the ultrastructure? The mitochondria is formed of a two limiting membrane. Outer membrane is with a less protein and freely permeable, but inner one is little bit denser one and it is semi permeable. Outer membrane is smooth and the inner membrane is produced inward and it forms cristae. And this cristae may be branched or sometimes may be unbranched. The matrix of the inner membrane, there is some series of regularly placed knob-like structure is present, which is known as oxysomes. And how it is look like? And these oxysomes are of three parts. One is base piece, stalk and head piece. I will show you the diagram. Then mitochondrial chamber. One is outer chamber and another one is inner chamber. Outer chamber is actually filled with a less dense medium, that is water, minerals and enzyme. But the inner chamber, it is filled up with a more denser and granular mitochondrial matrix, which contain different types of enzymes of protein and the lipid synthesis and Krebs cycle. Krebs cycle is present in respiration, that is plant physiology. You will get the Krebs cycle. Now what is the function? It is called the powerhouse or the battery or the ATP mills because it generates energy. Mitochondrial matrix has enzyme for elongation of, that is fatty acid chain. See at a glance the structure of the mitochondria. Now this mitochondria, this one is outer membrane and the inner membrane. Okay, outer and inner membrane, outer chamber and inner chamber. Okay, and these are the some structure, knob-like structure is present with a blue color pen. Just see the knob-like structure is present, is known as oxysomes, and this structure is known as crista or cristae, Cristae. Okay, so cristae is that is finger-like projection. Now see the alta structure. This one is alta structure, outer membrane, inner membrane. Outer space is a permeable and inner space semi-permeable. This membrane is semi-permeable and inner space. So this one is, that is one cristae. This one cristae with a knob-like oxysomes are there. This knob-like oxysome, how it is look like? One base piece, stalk and head piece. So it is forms the oxysome. The structure of the mitochondria and alta structure or the microscopic structure of the, that is electronic structure of the mitochondria is very, very important for all of you. Next one is our plastids. So the term plastid was given by the Haeckel and it is found in the plant cell, all of you know. And plastids are of mainly two types, leucoplast and chromoplast. Leucoplast is a colorless one and it is generally present in the root, rhizome and the seeds. And the chromoplast, it is a colored one and it is present in the part which are exposed to the light, that is leaves and flowers. This leucoplast again are of three types. Amyloplast, that is stored starch, that is in wheat and rice. Eleuroplast, it stored protein, maize grain. Then eleoplast, it is present in, that is castor seed and mustard, and it store fat. These are all colorless. Now this chromoplast is chloroplast, pheoplast and rhodoplast. This chloroplast green in color, green plastid present in the leaf. Pheoplast brown pigments, so algae, brown algae, and rhodoplast red pigments present in the red algae. Now what's the alta structure? First one is limiting membrane. These are two in number, and outer membrane has less protein and more permeable. Inner membrane more protein and semi-permeable. Stroma or matrix, it is denser and it is form of soluble protein with a ribosome, then naked DNA molecules, and it is a site of carbon dioxide fixation and protein synthesis. Then grana, inner, that is membrane, that plastidal membrane is invaginated to form a number of oval shape, a closed sac-like structure which is known as thylakoids. it is structural and functional elements of chloroplast. Now function, site of photosynthesis, stores starch, and for rest it helps in respiration. Now the structure of the chloroplast. So these are grana or granum. This liquid part is stroma, inner part is stroma. This single one thylakoids, inner membrane and outer membrane. This structure is also important for you.